Stanford University.
With that, it's a great pleasure for me to introduce Dr. Keller. I've known Gordon for a long period of time. I've considered him as a role model, as a mentor, and Gordon has made seminal discoveries, and I would really say seminal discoveries, and probably what we would call the grandfather in terms of the field of iPSL, ESL, in the field of cardiac differentiation, endothermal differentiation. He's somebody that all of us look up to, all of us asking for protocols, asking for advice on how they benefited quite a bit from his advice and interactions. So just by way of background, Gordon received his bachelor's degree in University of Saskatchewan in Canada. He then did his PhD in immunology at University of Alberta. And afterwards, he traveled quite a bit. He went to Basel, Switzerland, to be a member of the Basel Institute of Immunology, went to Vienna, Austria to be a visiting scientist at the Research Institute of Molecular Pathology, and then he went to Denver, University of Colorado Health Sciences as assistant associate professor, and then he went to Mount Sinai at the Department of Gene and Cell Medicine, also the director for the Black Family Stem Cell Institute. And then around 2007, he went back home to Canada to become the director of the McEwen Center for Regenerative Medicine. He's, as I mentioned, he's made tremendous contributions to the field. He's the editor for the journal Development. He was also a past president of the ISSCR. He's published more than 170 papers, pretty much all of them in high-impact journals: Nature, Nature Genetics, Nature Biotech, Cell, Developmental Cell, Cell, Stem Cell, PNS, and so forth. And more recently, he's co-founded a company called Blue Rock Therapeutics, in which the goal is to put pluripotent stem cells into patients. And I could go on and on, but I think you guys want to listen to him talk. So thank you very much, Gordon, for coming.
Yeah, you know what? It was making noise.
That's fine. I just did that for a time being.
Well, thank you for the kind introduction. And while you were talking, I was looking around and seeing all the young people in the audience, and I'm beginning to wonder what's going on here. And then you kindly referred to me as a grandfather, so I think we're knowing where I'm sitting. So it's a pleasure to be here and to see all the young people here, because you are the future. And I'm going to tell you a story today about a challenge we took on maybe starting 15 years ago or 10 years ago to make heart cells. And the goal evolved into getting over the fascination of seeing a beating cell in a dish, which, if you can wind back your clock 10 or 15 years, was pretty exciting then. Everyone sees them now. To being able to make all the major cell types of the human heart in a reproducible fashion and in purity. And we're not fully there yet, but we're almost there. And I'm going to go through with you the strategy showing how we've taken cues from developmental biology. I just spilt this on this. That's okay. You can buy a new one, Joe. And how we apply that to what we think is human development in the dish. So we're all familiar with this, where we take a pluripotent cell and we can make multiple cell types now. And from there, applications are ongoing in many of these different areas, ranging from cell therapy, disease modeling, drug discovery, and even teasing apart human development. And I think it's a wonderful opportunity, a great time to be getting in the field, because a lot of the heavy lifting of testing reagents has largely been done. So I think you can go in and ask really good questions now. So I'm going to talk a little bit about cell therapy and disease modeling. And I think for this to be successful, we need to think about two things. And always remember them. What is the target cell type of the disease, if you know it? And secondly, you need to then be able to make that cell in the dish to either study the disease or treat the disease. And it seems obvious now, but some years ago, it wasn't necessarily so clear that when you had beating cells, we'll just model disease or put them into an animal model and ask, will they repair myocardial infarction? And so I'm going to focus the entire talk on cardiac cells and touch at the end on these two aspects. But what I really want to talk about is how do we make these different cell types, and I want to show you how timing factors and reproducibility becomes a key point if you want to make these different cell types. So clearly, we know there are different chambers, and we came to this thinking that each of these chambers is colonized, made up by cells from a different lineage. These are specific lineages rather than just cells that can flip back and forth. That's how we approached it. So we have ventricular cells, we have atrial cells, and then we have the primary pacemaker of the heart. That's the sinoatrial pacemaker, another pacemaker. So those are kind of the major myocytes. And if we think already of what we might want to do with these, if we're going to repair myocardial infarction with cell therapy, we would want ventricular cells. If we're going to model, say, a disease such as atrial fibrillation, we'd want a pure population of atrial cells. And if we want to think about pacemaker disorders, we would want access to these cells. Complicating the issue more, of course, is there are surrounding layers that are fairly important in heart development and instructive. The epicardium, an epithelial sheet that surrounds the heart, and the lining, the specialized endothelial cells called the endocardium that participate in valve formation. And so the challenge, in retrospect, we didn't start with this challenge, but we developed it, is to see, can we figure out the developmental cues to drive each of these cells, and what can we learn from this that we can then feed back to developmental biology as our contribution rather than just taking from developmental biology. And so this is really what we've adopted many years ago, that there's been just a wealth of information in the literature on how the heart forms, and it's pretty consistent from animal to animal. So at least the major cues you can tease from that and ask these questions. And so if you look at development of the heart, certainly a mesoderm origin, these cells migrate to the anterior region where they form an epithelial crescent, which then coalesces into a tube, which then gains some identity where you already have ventricular and atrial populations in the tube. We get then repositioning of these cells on top of the developing heart and the four-chambered heart. So we're not going to end up today with showing you a picture of a four-chambered heart we made, but we will show you how to make most of the cell types. And it's really this type of approach, drawing from development and asking the question, can we reproduce this in the dish? So the challenge is, if we look at this almost as a hematopoietic diagram, here's our end stages, here's our stem cell, and this is what we have to fill. Now this differs from hematopoiesis in that it's an ongoing process. This is a developmental window where it happens, and then we move on. So I'm going to show you a lot of different lineage diagrams like this, but we've laid this out in a linear format where we look at a primitive streak-like population, we drive it to mesoderm, and then we have to pattern that mesoderm, specify it, and make our different cell types. And so we realized some years ago that we could use some of these markers, T brachyuria for primitive streak, MESP1, KDR, and PDGF receptor for mesoderm, NKX2.5 for our major indicator of cardiogenesis, and start asking questions. And this was back when Steve Kapman was still in the lab. He worked out this idea that patterning or induction at this step is exquisitely sensitive to these two factors. It requires Wnt as well, because I'm sure a lot of you who do this are using simply a Wnt-based protocol. There's a lot of endogenous Wnt, so we seldom activate it, but if you inhibit Wnt here, it is essential. But it's these two I'm going to come back to time and time again for this talk. We always measure our mesoderm induction, and I would strongly encourage anyone who's doing this to measure your efficiency early on, because if you're waiting 10, 20, 30, 40 days, you could have a disappointing outcome because you didn't make good mesoderm. You then have to inhibit Wnt. This is work with Mark and I were talking about this early on, that he's contributed to our understanding that to specify this to a cardiac fate, you need to inhibit Wnt. This is largely what we're going to talk about, and I know the details are a little bit more than you might want to understand, but it's really it's incredibly important, I think, if you want to make the right endpoint. We've used these combination of BMP and activin, and they vary depending upon what you want to make and your supplier. And then, of course, we can measure with various flow cytometry assays our success. And I think if we can bring more flow cytometry into this, we'll all be better off, because you can assay very quickly and quantitatively success and failure. So what are we making? So this is kind of a generic protocol. Many groups started with very similar protocols. Turns out if you look at MLC2V as one indicator, you can look at other markers. We're largely, by these early phase 1 or version 1 protocols, are making ventricular-like cells. And you can convert this to a flow-based, intracellular flow-based assay using troponin T as all of your myocytes, and this is your ventricular-like ones. We didn't even know we were making ventricular cells. It turns out we probably are. Patch clamping says these are most like a ventricular population. Any electrophysiologist in the audience will question whether this is correct. Absolutely mature, no, it's not mature. And there are also a few pacemakers in these populations, and the degree varies on experiment to experiment. And so as it turns out, we were making ventricular cells. And so we have kind of checked that box, at least for now. And so then we wanted to move on to atrial cells and ask, how do you make atrial myocytes? And again, the developmental biology field had done this understood this years and years ago, and others actually have done it as well before we did. But the goal was, how are these chambers specified? What are the cells that contribute to them? How are they different? Nadia Rosenthal and a few others have looked at retinoic acid signaling and did some very nice experiments on manipulating the pathway very early on. Showed that if you block RA signaling early, you lose atrial formation. If you activate the pathway above what would be normal, you enlarge the atria and you lose ventricles. So that gave us a pretty good clue. But most impressive is there was a window of opportunity, if you want, where if you missed it, you didn't really change the balance. And that told us this has got to be a very interesting developmental stage. And so using a number of markers, we used electrophysiology, but these are the IRX4 is a good transcription factor of our ventricular, MLC2V, and a suite of atrial markers. And again, you can extend this to as many markers as you want. And we simply then added RA. This is a really straightforward experiment, but it's one of those that worked the first time and gave very clean results. So if you look at the troponin T output, it's the same no matter when you add. But if you add the retinoic acid, this is all trans retinoic acid between day 3 and 5, you largely lose your ventricular markers, but you still have myocytes. And you're gaining atrial markers. And so a simple addition at that point tells you a lot. So here's based on flow cytometry. This is using an NKX 2.5 GFP reporter line to report the myocytes along with troponin T. You can see RA does not impact cardiogenesis, but you simply lose MLC2V positive cells. And electrophysiology tells us that these cells in the atrial cultures are quite different, and at least our colleagues say these are much more like atrial cells. And so if you look at this now, and I'll just take a minute to review what I said. This was our original protocol, giving a ventricular-like output, and if we add RA between 3 and 5, you get it to atrial. But if you wait till day 7, you lose that ability to make the cells. So it becomes an interesting experiment if someone's interested in what changes between day 5 and 7. Why do they respond to RA and make atrial at day 5, but not at day 7? So I think the biology you can do now is really impressive. You don't have to do more than just repeat this and start asking those questions, if those are the questions that interest you. But you have to wonder then, where does retinoic acid come from to do this? Because it's not just something that's floating around. This is a derivative of vitamin A that presumably comes through the maternal circulation. And this is really, retinol is then processed into an active form of retinoic acid. One of the enzymes I'm going to mention is retinol dehydrogenase. This is developmentally regulated. There's 3 forms of this. RALDH2 is the developmentally relevant form. And if you make too much retinoic acid, or any time you make it, you upregulate the enzyme that breaks it down, CYP26A1. There's a few other CYPs. These are P450 enzymes. So this is not a pathway that's going to be active for long periods of time. It's a very, very potent pathway. So Nadia went on to propose the following, that there's, the heart is made by cells that come in waves from the, and others have proposed this as well, from the primitive streak. The first wave contributes mostly to the ventricular makeup, and I'm not sure if we really know in her model if it's right or left. And around E75, you can map very nicely a region of RALDH2 positive cells. So these are cells that convert retinol to retinoic acid. And the model was that these are the atrial progenitors that contribute to this part of the tube. So you'd have the atria and the SA node cells. So this is the retinoic acid-rich region of the heart tube is making these cells. And so Cedric Blauplan in Belgium did an experiment that fits kind of with this, where he had a MESP1 tracer, and you could turn it on and off as to when you want to trace when the cells are coming out of the streak. So if you activated DOX early, you'd mark the cells early. If you activate it a bit later, you mark them a bit later. And what he found, if you mark early, you contribute largely to the left ventricle. If you mark later, the number of clones aren't terrific here, but it's suggestive you would make more atrial cells and perhaps right ventricular cells. And then outflow tract, of course, is made later as well. So there is this temporal type of distinction between the cells that contribute to the ventricle versus the atria in this experiment. But fitting with the whole retinoic acid story is he did a RNA analysis between the early and the late, and RALDH2 was highly upregulated in those late migrating cells, fitting with the notion that the cells that contribute to those compartments have the capacity to metabolize or synthesize retinoic acid. So we made a prediction that in this model, we should be able to recapitulate this. We should be able to have RALDH2 positive cells contributing to atria, if the model is correct. So we used an assay. It's readily available from Stem Cell Technologies. It's called the Aldeflor assay. And you have a fluorescent compound that freely moves through cells if they don't have any of the aldehyde dehydrogenases. And there's about 18 of them, of which RALDH1, 2, and 3 are 3. Once, if the cell is expressing, it's converted to a substance that cannot move through the cell, turns green, and you'll see it by flow cytometry. So it's not specific to RALDH2, but it is certainly measuring it if it's there. So here's our induction, and we're using here, for those of you at the back, this is PDGF receptor alpha. This is our mesoderm. And this is the mesoderm that would be showing this activity. So it's not really impressive with our protocol. So either the atrial cells are not coming through this, or we're doing a pretty bad job on making mesoderm that's going to make atrial cells. And so we asked the question, is this as good as we can do? And this is a pretty simple approach, but it goes back to what Steve Kottman had brought into the lab, is are we making the right mesoderm, or is there another mesoderm that may express more RALDH2? Or can we induce RALDH2, which is the other option? And I'm going to show you, this is published just recently. I want to show you, I'll go through just one of these because they're a little bit complicated. But what we did is we held BMP constant, just varying in those 2 days. And then we ran activin in a very shallow grid. This is for those at the back, 2, 4, 6, and 10 nanograms. And here's our Aldeflor positive population. It goes from 4% to almost 60%. We now know it's activin nodal signaling that's regulating either the enzyme that that reads on the assay or the mesoderm that expresses this. Now, the price you pay for this is we lost cardiogenesis. So then we held activin constant and modified BMP, and we recovered our cardiogenesis here. So we got 60% RALDH2 or aldophilor positive and almost 80% cardiogenesis. So what we think is, to synthesize this, we think that the activin nodal pathway is regulating the aldophilor activity, and let's call it RALDH2. And I can tell you we can say that because we made a RALDH2 null ES line, and it doesn't show aldophilor activity. So we believe it's all RALDH2. And BMP determines if it's cardiogenic or maybe something else. Somatic cells also express this. So that's kind of the balance you need. Now you could say, well, who cares? Does it matter? And so here's the original protocol we started many years ago, and here's the way we would do it now. And here's the gene to say, yes, the aldophilor is tracking with the expression of RALDH2. That's the gene. This is the optimized one for aldophilor. This is the less optimized. But I want to show you the enzyme that breaks down CYP is expressed very sharply and high in these cells. So based on this, we hypothesize that this could be atrial mesoderm and this is ventricular. And the ventricular mesoderm never wants to see retinoic acid, or you can override it and they'll become atrial cells. I think that's the balance you have in the embryo, and that's the balance that's at play in the dish. And so we sorted this cell, the positive ones here, and simply asked the question, do these cells have the capacity to respond to retinol? That would mean this enzyme is functional. And we sorted them, cultured them for 2 days, cultured them with and without retinol for 2 days, and then just let these little clusters go for another 18 days or 16 days, and simply ask the question. And what you're seeing here is they made cardiomyocytes very nicely. MLC2V isn't terrific from this mesoderm. It's a mesoderm issue, but you're making some ventricular cells. And they respond to retinol by losing MLC2V positive, and they, of course, respond to retinoic acid. And here, the atrial genes are upregulated. So these cells can respond to retinol, so we can check that box. And so we started bringing out a model that we believe that these two major cardiomyocyte subtypes are made from mesoderm that's specified differently, different populations right from the get-go. And I think this is fundamentally important if you want to make subtypes of cells for whatever your purpose. And so we asked then, what about a marker for mesoderm? And this was something we've been doing in the lab for a while. It's our version of a screen. We have about an antibody panel now, about 400 anti-CD antibodies just collected. It's just a mindless, you throw the cells on and ask, will they respond? These are all markers of maturing and mature blood cells. But we found, interestingly, glycophorin A, which is known best for being associated with red cell development, marking a subset of this cardiogenic mesoderm. And we just ran the antibody over our panel and found that these cells that express very little aldophilor have a lot of glycophorin A. These do not. And this is just a gene expression showing that glycophorin A is coming up. And these are mutually exclusive populations, so you can probably see where this is going. I would like to point out that not all the anti-CD235A antibodies work. There's one in our paper does, and we found one other, and we have one that doesn't, and we don't quite understand why. So this is the key experiment of this part, and we sorted these two populations. This should be should not be able to respond to retinol. This should. So here's the experiment, the three groups of the CD235A positive and the three groups of the aldophilor positive. And this is showing that they in fact do not respond to retinol in that they don't lose their ventricular marker. They of course respond to retinoic acid. So it tells you that you can make whatever mesoderm you want and throw retinoic acid on and get some ventricular cells. This shows that the green cells are the aldofluor positive ones. They do, I showed you, respond to retinol. Here's the similar data with the transcription factor. The atrial markers are the opposite. We get no response to retinol. We get a response to RA, and similarly here as well. And so we now have two populations and where we can now mark this and sort it with or monitor with glycophorin A. We believe these are ventricular progenitors, they're mesoderm, and these are atrial. And they're physically separate and they're patterned differently. And so if you want the best cells, our opinion is you need to make the right mesoderm. And so can we even get better ventricular mesoderm? I'm not going to go through the whole grid again, similar strategy, but indeed, we can now get a really impressive 235A positive aldofluor negative that gives us a very nice MLC2V readout. Now this type of analysis, these type of grids have to be done with each cell line you start with. You can't use the same grid for 6 different cell lines. You will see it doesn't work. And even more worrisome is you have to do it each time you get a new batch of activin or BMP. I'm just the messenger. I'm not the producer of these reagents, but that's really where we're at now because these are very shallow concentration changes, and so we're looking at any probably problems or changes in the absolute activity will impact this. So for this part, this is really the message I want. We believe that we have a specification of a population that makes ventricular cells and a population that makes atrial cells. These can respond to retinol. These cannot. These, of course, will, in the presence of high amounts of retinoic acid, come down here, and these will make some ventricular cells. But by and large, we believe this kind of recapitulates what the models in the mouse told us, that we have different cells contributing to different parts of the heart. So what about the SA node? And I'm trying to weave this all together so when you think about this, you can say it's not, we can't figure it out, we don't know how it all fits together. And I told you already where you have a suspicion where these are going to come from. This we published a bit earlier this year. And in this case, the SA node comes from this sinus horn, and upon looping, sits on top of the heart. And this is work that Stephanie Prot's postdoc in the lab carried out, and the former work was hers and Ji-Hun Lee, a student in the lab. And the clue came from the fact that this is a section of a human fetal heart, which shows very nicely that the nodal region is NKX 2.5 negative. It's probably one of the few components of the heart that does not express this transcription factor. And so Andrew Elefante and his colleagues kindly sent us his reporter line. So we had a reporter for NKX 2.5 under GFP, and we used the marker that we identified, SERP-alpha, on myocytes. So the hypothesis is not a deep one. It just says that we believe that the NKX 2.5 negative population is the one that's going to have the pacemaker cells. And just to position you, this is now all coming from that atrial mesoderm type population because that's where we believe the pacemaker should come from. And this was pretty straightforward. We saw there was, obviously when you drive this differentiation with this reporter, you have NKX 2.5 positive cells that have some troponin T. So there was our hint already. And there are the two populations, and if you just sort and look at them, and I don't know if you can see this, these are the positive cells beat quite slowly, and these beat much faster. And that already gives you a clue that these two cell types are different. Obviously, if you mix them, these are going to drive these, and your whole culture beats faster. That's why most of the cultures beat quite fast. If you strip these away, these always beat much slower. If you look at transcription factors, the way this is set up, this is our NCAX 2.5 positive. This is our putative pacemakers. This is fetal ventricular from fetal tissue. This is fetal SA node, fetal AV node. TBX18 is known to be expressed in the SA node, so is this transcription factor SHOX2, islet 1, and TBX3. The top ventricular-like cells are MLC2V positive, ours are not. They're RX4 positive, ours are not. And a key experiment here is to show that the cells are TBX2 negative. So this is an AV marker. We propose then that these are the non-pacemakers, ventricular-like, maybe atrial progenitor type, and these are putative pacemaker cells. Electrophysiology would argue that they're different than the ventricular cells. And this is the key experiment. And this was, for those of you who you know, a story about collaboration. And this was when Leor Gepstein was in the lab on sabbatical. He said, you know, the only way you're really going to convince someone is put these into a heart. And so what he did is he said, we can send them back to Israel, and my student can put them in the apex of the heart for 10 to 12 days, and then we'll put them out on a Langendorf chamber and ask, can they pace? And so we took him up on this and made these cells, sent them to Israel. This is the heart. This is where the cells were injected. And this is now just a sinus rhythm you're looking at. So this is beating very fast. The movie slowed down, but the pulse is coming from here. And you can do a color map to show that indeed it is not coming from where we injected the cells. Now you can almost induce a chemical AV block where you slow the heart down, and now you can see this, the signal starts here. And you can see it coming from here. And so we've done a lot of work on histology sections and that to show that there are indeed human cells down there. And so if you then look at it in this pattern where you're actually looking at the electrophysiology, this is the rat heart beating. This now switches to the human taking over, and when the drug wears off, back to the rat. And you can see the color map shows very nicely. This is with the SA node cells. The ventricular cells are unable to pace. It kind of went into complete heart block and then recovered. And so we use this to suggest that at least in this model, it's out there in the sense that it's only for a short while this is happening, we're actually getting pacing. So what regulates development? And this again, I'm going to go through this a bit quicker, but really this came from work on developmental biology, that this is the region where we get two populations, the proepicardial organ that makes epicardium and the pacemaker. And we had previously shown that to make proepicardial organ cells, you have to hit with BMP4. So we thought BMP4 was a good shot given the proximity. And to make of course I talked already about the atrial cells and the RA gradients, that RA would be dramatically the number of pacemakers, and it's up probably double or triple, and there's again a window of opportunity. So we activate BMP after you make the mesoderm, and you get more pacemakers. Retinoic acid, very similarly. In this case, retinoic acid doesn't increase the proportion, but it alters the gene expression. So same number plus minus retinoic acid, but as soon as you have retinoic acid, the expression of the pacemaker genes increases. So it's having a bit of a different effect. And the beating rate also goes up because they are pacing. And the final one that really allowed us to move this away from the NKX 2.5 line was the observation in the literature that FGF signaling is essential to turn on NKX 2.5 or to induce this population. And so now we did an FGF gradient, and here you can see timing again, or you can see the effects, control, plus FGF, minus FGF. We skew. And so to add to this graph, this is really what you're looking at now. So instead of going this way, you add BMP, RA, and FGF inhibitor, and look at the NKX 2.5 negative, and you have your pacemaker. And this is the final. This has been published some years ago, but I want to round up the story because I think it's hard to put it all into perspective. It's the epicardium. This is work that a student, Alec Whitty, did for his PhD. And it's not a lineage we think much about, but it's incredibly important. It comes from a proepicardial organ, and these cells literally crawl around the heart and surround it. And it's identified by TBX18. You can see RALDH2 again. Some of the cardiac genes are expressed in this epithelial population. And so a key source of cardiac fibroblasts, and this is really what we would like to do, is generate cardiac fibroblasts from these cells. And so here we manipulated BMP and Wnt at this stage, and we ask, how does it work? And I'm going to just summarize because this is published. BMP minus Wnt gives you cardiomyocytes. If you allow Wnt to be active, when Mike or Chuck have isolated cells or looked at the cells in the graft, they're predominantly ventricular. But we don't know, they put 1 billion in, 10% survive and something grows out. So is it a selection for ventricular cells, or was it conversion, or did they start with a pretty good population? With highly enriched populations, you can address those questions a bit better. Yeah. No, I think it's across the board. They're largely immature cells, and I think we're missing, we're focused, we're maybe thinking too much as early developmental biologists, and we're not bringing into account maybe things that happen at birth, the dramatic change in hormones and oxygen tension, the change in metabolism, huge for the cardiomyopathy, has to be factored in as well. I hope someone else can figure that one out. Yeah. 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 Right. They largely, they didn't have the resolution on the mesoderm subtypes. They had cardiac mesoderm, blood mesoderm, and I think periaxal, I think. I can't remember anymore. Yeah, I mean, at that level, I think they made some interesting observations on how you can drive these with small molecules and what other factors you have to add. But now we know, and we published some years ago, that blood cells also come from separate mesoderms. So the next iteration of that approach has to be taking into consideration that there isn't just one type of blood mesoderm. There just isn't one type of cardiac mesoderm. It's complex, and that's just the way it is. The question we have is how different is the blood and the cardiac mesoderm we're making? Because primitive blood cells and ventricular mesoderm both have CD235 on them. So are they the same? Are they different? We don't know.
One last question.
Yeah. Yeah. Yeah, the SA node, it's so for the SA node, that patterning step was D3. All of it's based on timing and developmental biology. Okay, thank you very much. Thank you.
Stanford University.